Hi everyone, my name is Powell and I'm a research fellow with the Department of Anatomy with Radiology at the University of Auckland. In this talk, I'd like to show you some of the tools we've developed to analyze the shape and function of the heart. First, let me decipher the title for you. An atlas is a collection of maps. In this case, maps of the cardiac anatomy. We want to see how these maps change between normal and abnormal patients. In this case, patients with myocardial infarct. These maps will account for the shape variation in space and through time in a continuous way. In my group, we mostly use MRI. It is remarkable how much information we can acquire when we put a patient through the MRI scanner. Typically for a patient we obtain hundreds of images which we can animate into movies of the heart cycle. By fitting mathematical models, as we'll see, to these images, we can derive clinical parameters such as left ventricular mass, end-diastolic and systolic volumes, wall thickness, ejection fraction, etc. These are used by clinicians since they correlate well with certain cardiac conditions. But what about the rest of the shape information? For example, can we help clinicians identify patterns of shape and function variation which are related to disease? To be able to separate normals from pathologicals, we need to first define a reference of normality. And to do this, we need an alignment or registration procedure to be able to map features across different cases. Many approaches have been proposed for this problem in the context of cardiac modeling. Some references are shown here, and their details can be found in my thesis. The Cardiac Atlas project was set up to establish a structural and functional atlas of the heart. This project is dedicated to combine cardiac modeling and biophysical analysis methods with a structural database for the comprehensive mapping of heart structure and function. We have collected more than 2,500 de-identified cardiac patients along with the corresponding 3D finite element models. Researchers can therefore apply to access the cardiac data for specific research projects, and clinicians are encouraged to contribute new cases. For this work, we use 300 cases from the Determined study. These are patients with myocardial infarct and represent our sample of pathology. From the MESA cohort, we used nearly 2,000 models. This is a subclinical study, which means a study of the progression of disease, of atherosclerosis, but at the time of recruitment the patients presented no heart-related symptoms, and we therefore consider them normals. To create this atlas, we're going to have to deal with several problems. How do we register or align all shapes to a common template? How do we align the models in time? The second question is going to lead us to find a continuous interpolation. To align everybody to the same map or template, we use customized software to fit a finite element model of the left ventricle to the MRI images. This is done in a standardized fashion and provides a mathematical representation of the endocardial and epicardial surfaces throughout time. By using some fiducial markers at key anatomical positions, point correspondence within a small tolerance is guaranteed. The finite element model is written in prolate coordinates by means of Bezier parameters which capture the specific shape of each patient. This fitting procedure was applied to a subset of the MESA sample for validation purposes. However, the rest of the sample was automatically fitted from drawn contours by experts. This automatic fitting procedure accounts for the misregistration which arises from breath hold during the MRI scan. Validation experiments showed excellent agreement between the automatic and manual approaches. To analyze the shape parameters, we mainly use principal component analysis, or PCA. This statistical tool finds patterns of variation within the shape parameters by maximizing the directions of variance. Let's look at an example. Let's say we've got a classroom with several students, and each student's height and weight has been recorded and is shown in the figure. If we look at the distribution of the cloud of points, we can see that there is a direction of maximum variance, which is represented by the major axis of the ellipse in red. This direction is related to size, and it's telling us that people who are taller tend to be heavier. The second orthogonal direction, or minor axis, is telling us that there is some residual variation, not explained by size, which is primarily related to weight. This is telling us that for any given height, some people are heavier than others. We call this mode sphericity. Each one of these directions are sometimes called eigenvectors because they are the product of factorizing the covariance matrix. In linear algebra, these vectors and their associated values are termed eigenvectors and eigenvalues. We've seen thus far how we can derive models from the images, be it automatically or semi-automatically using the guide point modeling. However, before we can compare a set of shapes, we must ensure that differences due to position, orientation, and sometimes scaling are removed. Procrustes alignment is very helpful in this regard. If we think of our hearts as the blue stars, the Procrustes method aligns all the blue stars to a common position and orientation represented by the white star. And if you wish, it can also remove scaling in the process. As you can imagine, certain shapes, especially symmetrical, will have multiple mappings to that common white star. Fiducial or anatomical markers ensure the correct orientation for each shape. If we do not include scaling in the Procrustes alignment, as in our previous example, size is the dominant mode, explaining almost half of the variation at end diastole and end systole. The animations show different weightings of the mode, from -3 standard deviations to +3 standard deviations, virtually covering the whole population. If we, however, remove scaling, the second mode of variation at end diastole and end systole is, like in our previous example, a sphericity. This component explains how much a heart resembles a rugby ball or a soccer ball. What we've seen until now was static, that is, analyzing a time frame at a time, ED or ES, independently. Our shape parameter looks like the B vector shown here, containing the Bezier parameters of our finite element model at a given time. If we wish to analyze these changes temporally, we need to align the different beating hearts and time so that they all beat at the same speed. This is done using a piecewise mapping which ensures that all hearts are at their minimum and maximum volumes at the same time. If we look at one shape parameter over time, we can see how it varies between the now normalized time axis. Since we wish to find a continuous representation of this function, and since this function is clearly cyclical, we used its Fourier representation with 5 harmonics to contain the number of dimensions of the shape vector. Now, the shape vector B contains nearly all spatial and temporal information in an aligned or registered way suitable for PCA. Now, we applied this spatio-temporal PCA to 300 MESA and 300 determined cases and compared them. In this example, we have removed scaling as well, and you'll see that size has disappeared. In the MESA or asymptomatic modes of variation in figure 2 on the left, it could be reasoned that the first mode corresponds to the lengthening component of the ventricle, and modes 2 and 3 correspond to features of the mitral valve geometry and base plane tilt. However, from an overall geometric or clinical perspective, in this case, there are no pure modes of variation, for example, just sphericity. In the determined or asymptomatic modes in figure 3 on the right, mode 1 represents their sphericity, as was also found in a previous static analysis. Mode 2, the lower midventricular thickness, and mode 3 shows mitral valve geometry features along with a rounding or bulging of the apical region. These features correlate well with clinical indicators of heart failure, that is, sphericity, wall thinning, and local dilation of the ventricle, which are all features of infarcted models. Unfortunately, my PowerPoint crashes when I try to play too many animations at the same time, so these modes are animated online in our website, and I would like to invite you to explore the rest of the modes of variation. Sometimes it can be a challenge to label them, since when they lose power, the variations become subtle and more difficult to identify. In conclusion, we have seen an approach to register a bunch of models in time and space making use of a number of tools. In this talk, we've presented a compact representation of the spatiotemporal variation of the left ventricle and have shown an application in both normal and infarcted patients. We have also seen how some of these modes relate to clinical indicators of heart failure. By using these differences between modes, we open a door to automatic classification, which we have and continue to investigate. Thank you for your attention. I hope you're still there. I'd like to thank my supervisors, Alistair and Brett, and my colleague Alvin. I would also like to thank the expert analysts who fitted the models, Agustin and Ben, and our head programmer, John. If you'd like to learn more about any aspect of this presentation, here you'll find some links to some of our publications. I hope you enjoyed it, and I might see you at Mackay. Come and see some of these tools applied to the construction of a coronary artery atlas at poster number TP14.